Hey everyone, in this video we'll be discussing about pancreas. So what are pancreas? Pancreas are nothing but heterocrine or composite glands. So what are heterocrine or composite glands? So those are the glands that can function both as endocrine and exocrine glands. As you can see, pancreas have a pancreatic duct. Now pancreas are flower-shaped organs that are present in the abdominal cavity, and they are present right beside the small intestine. So as you can see, this is the duodenum, and duodenum is the first part of the small intestine. That is the first initial beginning point of the small intestine, and it is a C-shaped organ. So pancreas are present right in between this C shape of the duodenum, and here you can see it has a duct which actually merges with the bile duct of liver, and it goes into the duodenum. So this duct shows that it has an endocrine part. This small granular structures that you can see on this pancreas, these are nothing but the acini. So acini actually are the ones that perform the exocrine function. So they produce enzymes such as amylase, lipase, proteases, etc., and they go through this pancreatic duct into the small intestine. However, there is also another part in this pancreas that functions as the endocrine part. Here, like, there are small spots present on the pancreatic glands. So these are nothing but the islets of Langerhans. Now these islets of Langerhans, they have some cells called alpha cells, beta cells and delta cells. Now delta cells actually have a very different function. They are not involved in glucose homeostasis. Now this endocrine part of the pancreatic tissue, this what it does, it helps in glucose homeostasis, that is maintaining a healthy balance of glucose in our blood. So only two cells, alpha cells and beta cells, are actually involved in this glucose homeostasis. We'll be studying about the hormones they produce and the functions that they perform. So let's get started. So as I said earlier, these pancreas are nothing but composite or heterocrine gland. They function both as exocrine and endocrine glands. The exocrine gland performs digestive function, and the endocrine part performs the glucose homeostasis, which is very important to us in this chapter. So as I said, it has islets of Langerhans, the endocrine part, and they are 1 to 2 million in number, and they constitute a very small percentage of the pancreatic tissue, that is about 1 to 2%. And they have two main cells, that is the alpha cell and a beta cell. So first we'll talk about alpha cells. Alpha cells secrete a hormone called glucagon. Now what is glucagon? Glucagon is a peptide hormone. As I discussed earlier in the thyroid gland video, we talked about thyroid hormones. Now those thyroid hormones had a basic structure that was made of amino acids, that was tyrosine, on which iodine atoms were attached. And I said that tyrosine is an amino acid, and amino acids are the basic parts of the protein. So they are basic component of protein. So here similarly, this peptide is made up of amino acids. So this is a little bit higher level than the amino acid. So 10 amino acids, when they join together, they form a peptide. So glucagon is a hormone that has a peptide structure. And we'll be studying about peptides, amino acids, proteins, carbohydrates, glucose, etc. in detail in a chapter called biomolecules, where we learn everything about the structure of the organic molecules that are present in our body. This glucagon is a hyperglycemic hormone. So what do you mean by hyperglycemic hormone? It increases the glucose levels in our blood. So it increases and helps in maintaining that increased glucose level in our blood. And how it does that is, it acts on the liver cells. Liver cells or hepatocytes, they are called. And it promotes glycogenolysis. So what is glycogenolysis? Glycogenolysis is nothing but it helps in breaking down the glycogen in our liver. So glycogen is nothing but it is a storage carbohydrate in animals. Like how you have starch in plants, we have glycogen in our body. So whenever glucose is present in our blood, so whenever we eat food, it gets digested, we absorb glucose. When we eat excess of food, or if there is excess of glucose in our blood, so some gets used up for the functions of our body. Then the excess of the glucose gets stored in our liver and muscle cells as glycogen. So that is a glycogen for further use whenever we don't have food or when we don't have sufficient energy in our body. This glycogen gets used up. When there is excess of glycogen stored, it gets converted into fat. That is why in some patients they have fatty liver. So what happens is this glycogen gets stored in the liver cells. Excess glycogen keeps getting stored, and what happens is that glycogen gets converted into fat. So whenever we are hungry for longer times, like whenever we are fasting for 12 to 18 hours or so, this glycogen from liver cells gets broken down into glucose and our body starts consuming it. So this is one way that this promotes the glycogenolysis. So it helps in breaking this glycogen down and converting it back into glucose so that the glucose levels in our blood can increase. So it does by that, and it does it by one more way, that is gluconeogenesis. So what is gluconeogenesis? Gluco means glucose, neo means new, genesis means formation. So formation of new glucose molecules. So how it does it is by it helps in breaking down non-sugar precursors. So as I said earlier, our glycogen is nothing but a carbohydrate, right? So it is a carbohydrate. So this is the sugar and it gets broken down into glucose. So glycogenolysis. So glycogen lysis, that is lysis means breakdown, breaking down of glycogen. However, in gluconeogenesis, what happens is it breaks down non-sugar precursors. So what are non-sugar precursors? Your fats, proteins and so on. So it breaks everything that is not a carbohydrate, that is fats and proteins. We majority in our macronutrients we have carbohydrates, proteins and fats. So these are the ones that provide energy to our body. So apart from carbohydrates, these are the ones that are non-sugar precursors. So from this it converts them into glucose and it increases the blood sugar levels. So if you are thinking how can you convert fats and proteins into glucose, the structures of all these are very similar, the carbohydrates, proteins and fats, which we'll be studying again in biomolecules. So like they change or contort these molecules into glucose. And it maintains, as said earlier, it increases blood sugar levels and it also maintains those increased blood sugar levels. So how does it maintain that? So it reduces cellular uptake. So as said earlier, glucose when it comes into our blood, it goes to different cells. It helps in performing functions by providing energy. So what it does not let that glucose to be taken up by these cells. Like for whatever cells they have to perform functions, it prevents them from taking up this glucose and utilizing it to perform functions. So it keeps the glucose levels as it is. As I said, it is a hyperglycemic hormone. Hyper means high, glycemic means sugar. So it increases the blood sugar levels. So some of these are very important terminologies. So it is helps in glycogenolysis. Glycogen means breaking down glycogen, and gluconeogenesis means breaking down of fats and proteins, that is non-sugar precursors, into glucose. So gluco means glucose, neo means new, genesis means producing. So producing new glucose molecules, and in turn it helps in maintaining hyperglycemia. Hyper means high and glycemia means sugar. And also it maintains that high sugar levels by reducing the cellular uptake and utilization to perform vital functions. Now we'll be talking about beta cells. Now another part of islets of Langerhans are the beta cells. So beta cells secrete insulin. So what is insulin? Insulin is another peptide hormone. So it is very similar to glucagon in structure. It is also a peptide hormone. And this does the opposite of what glucagon does. So glucagon, what it does, it increases the blood glucose, it maintains the increased blood glucose. But insulin decreases blood glucose and it helps in utilization of the blood glucose to perform certain functions. So what it does, it regulates the sugar homeostasis. Again, homeostasis is a healthy balance we discussed earlier during hypothalamus. And it acts on hepatocytes and adipocytes. So what are adipocytes? Adipocytes are nothing but your fat cells. So fat cells that are present in the subcutaneous tissue or the visceral tissue, that is adipocyte. So it acts on the hepatocytes and adipocytes and promotes cellular glucose uptake and utilization. So as I said earlier, hepatocytes are nothing but the liver cells. These are the fat cells. So this is the liver cells and these are the fat cells. Sites means cells. So this acts and promotes cellular glucose uptake. So as I said earlier, glucose when it is present in excess gets converted into glycogen. How it gets converted into glycogen? This hormone promotes that conversion. So as I said earlier, it helps in promoting the conversion of glucose into glycogen. And this decreases the blood glucose. What happens when it decreases the blood glucose? It has to make this glucose go somewhere, right? So what it does, it helps this to convert this glucose into glycogen, and this glycogen gets stored in the liver and muscle cells. However, when there is excess of glycogen, it gets converted into fat, and this goes to the fat cells or adipocytes. So it does whatever it does opposite of glucagon. So insulin and glucagon together, they maintain blood glucose homeostasis. In next video, we'll talk about how the food is digested and the byproducts of the food produced, and how this glucagon and insulin work together to maintain glucose homeostasis in detail. Thank you.